Hello everyone, my name is Ian McLaughlin and I have a PhD in neuroscience, and today I wanted to talk about a pretty spicy scientific article that was published in the very highly regarded journal Nature just a few days ago that basically explores one of the kind of fundamental tenets of evolution by natural selection, like the way it works, the way we think it works at least. Are mutations really random? And so, I'm sure at this point most people are at least familiar with the concept of evolution, right? Organisms change over time. That's the short story. But I'm not going to, dive too deeply into the basics of evolution. But the basic idea is that genetic mutations occur somehow, and sometimes those mutations are beneficial. Sometimes mutations are detrimental, and sometimes they don't really have much of an effect. And those mutations are either inherited or not as organisms reproduce, right? If those mutations are inherited, then those mutations are passed through subsequent generations. If they're not inherited, they're not, right? And of course, there's a particular term that describes the process by which mutations are either passed down or they're not: natural selection. And so the story is obviously more complicated than what can be explained in just 4 sentences, but that's the gist. And a pretty big part of that whole process, arguably like the central ingredient to that whole recipe, is mutation. Without mutation, there would be no evolution. Mutation is essential to how species adapt to changing environments over time, to how viruses evolve to evade immune systems, and how new species reproduce. And new species emerge. So without mutations, right, there would be no Homo sapien. Our big old fancy brain and our helpfully opposable thumbs wouldn't have come about. But why do these mutations occur? Or more specifically, when it comes to this paper in particular, how do they happen? Why do certain genes mutate? Well, the sort of accepted dogma, I guess, so far has been that mutations occur entirely randomly, that essentially any gene within the genome is equally susceptible to mutation, that literally any gene in the genome is just as likely to undergo mutation as just as much as any other gene in the genome. There could be a mutation that occurs in a quiet, unimportant part of the genome. It doesn't really have any practical consequences whatsoever, right? And that's just as likely to happen as a mutation in a part of the genome that influences like how many fingers you have, right? That mutations are 100% random and unpredictable. That's basically the standard understanding of how mutations drive evolution by natural selection. The paper I'm going to talk about today, this paper adds a bit of complexity to that understanding. That it may not be the case that every single part of the genome is equally likely to undergo mutation. Rather, some genes are quite a bit more likely to undergo mutations than others. But before we go too deep, let's all get on the same page here. And before the end, we'll explore how it is that this whole dynamic can help understand how it is that new variants of viruses can emerge. So you might remember from your high school biology class that the whole pathway is DNA in your genome is turned into RNA, which is then turned into ultimately proteins. And that our genome is just a very specific sequence of A's, T's, C's, and G's. DNA, those A's, T's, C's, and G's, is transcribed into RNA, which is then translated into amino acids, which then fold up into various proteins that make up our bodies, right? And that's true. That's still true. This paper does not challenge that. There's no one who's seriously challenges that pathway. But there are opportunities for mistakes to happen, or mutations, all along that pathway of DNA to RNA to proteins. Okay, and so you're looking at 2 different examples of DNA being processed. One is where DNA is being replicated to new DNA, like when new cells are growing, like when your body makes new skin cells, for example. That one is on the top, and then the one on the bottom is when DNA is being turned into RNA, that process called transcription. And so, as you can see, this is a pretty frenetic process, right? With a whole bunch of molecules interacting in very, very precise ways, landing in very specific parts of the cell, interacting with very specific collections of other molecules, right? And so you can imagine mistakes can occur during, as these processes happen, right? The wrong molecule can land in those locations, resulting in a slight change in the resulting product. So instead of an A, you can get a T or a C or a G, which then can change the gene that ultimately, or the product of that gene, right? But there's a big difference between the consequences of exactly when and exactly where those mistakes or mutations occur. And so if a mutation occurs in one of those new skin cells, an A is replaced by a G, right? Generally speaking, you'll never even really know, right? Either it doesn't really make a big difference in the resulting protein, or it changes the protein so significantly that it no longer functions properly, and we have multiple cellular systems that are literally devoted to detecting and responding to proteins that are misbehaving in whatever way. Then biochemically flagging those misbehaving proteins for either being corrected or just being destroyed. Or, if none of those fail-safes kick in, you can have a cell that's capable of unbridled replication, right? And so it can just divide and divide and divide and just starts this whole process of completely uncontrolled cellular growth. And that process is what we call cancer, right? So that can happen in skin cells, for a whole variety of reasons, but probably the most intuitive is the fact that, our skin is exposed to radiation from the sun on a very regular basis. And some of us, or at least those of us who spend extra time in the sun, are a bit more susceptible to those kinds of genetic errors occurring in our skin cells, which is why we should all wear sunscreen when we know we're going to be spending, time in the sun. You're just reducing the number of opportunities that one of those errors will occur because fewer cells and the genetic activity inside those cells will be exposed to radiation that can cause those genetic errors to occur. Okay, so similar principle applies to other cells in our body, right, that aren't exposed to the sun. Lung cells. Well, those errors in, genetic activity can occur no matter what, right? But if you happen to smoke cigarettes, which introduces a whole bunch of highly reactive molecules, you're just introducing a massively greater number of opportunities for those genetic errors to occur, right? The byproducts of tobacco combustion, burning a cigarette, are super highly reactive molecules, some of which can interfere with all of that frenetic genetic activity, potentially, influencing that genetic activity in a way that results in uncontrolled cellular replication, right, and then generating lung cancer. Same idea throughout the body, right? Cells don't have to be exposed to things like the sun's radiation or to the radioactivity of a nuclear bomb or something like that, or from the smoke to the smoke of cigarettes. Or pollution in general, right? Those errors in the absence of all those environmental, exposures, mutations can still occur. But those environmental exposures just crank up the likelihood that those mutations will occur. But everything we just discussed are examples of mutations that occur when your body is already pretty much developed, right? So you're walking around pretty much fully rendered body, right? So what happens if those mutations occurs earlier on in our development, right? What happens if, for example, there's a mutation in the genes present in an egg or sperm cell, right? Well, the consequences of that are very different. Egg cells and sperm cells, have to grow while we're still sort of developing, right? It's not like someone just sort of crams all those eggs and sperm cells into our bodies after puberty, right? Those cells grow and develop, and just like in those, the rest of the cells of our body, there are opportunities for genetic errors to occur. And so, if there's an error in one of those cells, which is quite a bit less likely than in, skin cells, for a whole variety of reasons, but if there's an error in one of those cells, well, that error will be transferred to whatever organism results from the byproduct of the combination of that egg and sperm, right? With another egg or sperm cell. So the resulting offspring will inherit that error, that mutation. Those kinds of mutations are called germline mutations, and it's those kinds of mutations that are really what pushes evolution. And so as the lead author of the study, Gray Monroe says, quote, "DNA is a fragile molecule. On average, the DNA in a single cell is damaged between 1,000 and 1 million times every day. DNA also has to be copied each time a cell develops, or divides, which can introduce copying errors." End quote. So that's what we're really talking about here, right? Changes that arise in the DNA, in our cell's DNA for a whole variety of reasons. And there are other instances, if inheritable mutations, there are other ways that inheritable mutations can occur, and epigenetics, has introduced this whole additional kind of venue for inheritable mutations. But let's just stick with that model, right? A mutation occurs while an egg or sperm cell has grown up in somebody's body, and then that mutant sperm or egg is involved in a fertilization event that then results in a new human. Mutation, evolution, right? And so now, hopefully we're on sort of a similar page here. Let's get to what this team did. And so what it is that this team found that was surprising enough to rattle our understanding of how it is that mutations fuel evolution is that they may not actually be random. So first of all, this team was working with a rather adorable, fairly simple model organism, the thale crests, or Arabidopsis thaliana, or Thaliana, not sure. So, cute little flower, right? And you may be thinking flowers are nice and everything, but they're at least somewhat simpler than the human genome. And that's true, you're right. But depending on the question that you're asking in science, right, having a simpler model organism, like a flower rather than a whole human, can actually be better than studying a whole human. For one thing, humans don't necessarily always cooperate. We have things like moods, good moods, bad moods, silly moods, sad moods, right? We have appetites that aren't, necessarily entirely predictable. We have interactions with other humans that can make us happy or sad or angry or mad, right? And on top of all of that, we have a pretty expansive genome. 3 billion base pairs, 3 billion A's, T's, C's, and G's. And so if you're trying to understand how changing one gene in particular might influence human appetite or mood, right, working with humans would be great. But, of course, it's pretty tough to do that kind of research with humans for a whole variety of reasons, right? But if you're only interested in understanding how it is that genes undergo mutation over time, working with humans could be an incredibly cumbersome and unproductive approach, right? Not only do humans have all those 3 billion A's, T's, C's, and G's, but we also live a pretty long time. And, we tend to only have a few offspring, if any. And so, if you're working with humans on trying to understand the mechanics of mutation that drive evolution, you're going to have to follow multiple families, hundreds, thousands of families over the course of many, many generations, just so you can capture the genomes of parents and then their offspring and then their offspring and so on, right? And so other organisms can reproduce far more quickly than humans do, right? Multiple animals, even mammals do, right? But if your question, like the fundamental question of this study, is laser focused on answering one specific question, is every single gene in the genome equally likely to mutate in subsequent generations? If you're laser focused on that one particular question, you can attack that question far more effectively with almost any other organism than humans, right? What you want is an organism that reproduces very rapidly, an organism whose biology isn't, affected by a ton of things you can't control. Like what it's eating and when it's eating it, whether it just broke up with the girlfriend or something, right? Or if it only got 2 hours of sleep the night before, right? That kind of thing. You also want an organism that doesn't have billions and billions of A's, T's, C's, and G's, on which you're not focused. So ideally, you want an organism with relatively few base pairs in its genome, like, for example, just a bit over 100 million base pairs. So, guess which organism checks all those boxes? This cute little flower. And so this little puppy only has 120 million base pairs, right? That's not a massive amount of genetic material that you have to wade through to find your signal. It's not a ton of noise. It obviously reproduces pretty quickly, and it doesn't have a whole wide variety of moods, like my 3-year-old daughter when she doesn't immediately get the jalapeno chips that she wants. But so they're working with this thale crest, right? It's a good model organism, and they're trying to see if, as the plant reproduces, mutations occur totally randomly along its entire genome, or if those mutations actually occur according to some kind of a pattern. And so even though, they were working with this simple little plant, the study still took 3 years to conduct. And so they were growing, these hundreds and hundreds of plants in the lab over 3 years, and they ultimately sequenced 1,700 genomes. In other words, they figured out the specific sequence of the plant's A's, T's, C's, and G's, right? And so the table is set, right? So do you think we'd be talking about this paper if they just confirmed that the genetic dogma that every gene is equally unlikely to mutate? Of course not. This group found that mutations appear to happen non-randomly, that there's a pattern of some kind to which these genes tended to undergo mutation. And some pattern that made it so that certain genes were more likely to mutate than others, right? And so as that lead author of the study says, quote, "In genes coding for proteins essential for survival and reproduction, mutations are most likely to have harmful effects, potentially causing disease and even death." Duh. Our results show that genes, and essential genes in particular, experience a lower mutation rate than non-gene regions in Arabidopsis. The result is that offspring have a lower chance of inheriting a harmful mutation. That makes sense, right? That doesn't necessarily challenge, the kind of central assumption that every gene is just as likely to mutate, right? If a gene that regulates the formation of the valves of your heart mutates, that could far more likely cause a fetus to stop developing because blood isn't, effectively being distributed throughout the whole body, right? Whereas if a mutation occurs that makes your hair slightly darker, who cares? No big deal. Doesn't really affect you. And so, that's just good old natural selection. If a mutation prevents an organism from reproducing, or just makes it harder for that organism to reproduce, that mutation is far less likely to be transmitted intergenerationally, as a result of that mutation, right? Because it's just harder for the organism to reproduce, or its progeny to reproduce. But this group seems to have found that it's not even that simple. That, rather than any point along the genome being just as likely as any other point to mutate, there seems to be some relationship between the specific sequences of A's, T's, C's, and G's that are present at any point along the genome, and the likelihood that those points will undergo mutation. So, just like we'd expect, there are a few mutations in the parts of the plant genome that were fundamentally critical to survival, right? But they focused entirely on what are called de novo mutations, right? So de novo mutations, which is literally the Latin term for new mutations, those are the mutations that happen for the first time in an organism, right? That they weren't inherited, they just happened, right? This first time that a mutation has occurred. And so, at least for this organism. And so this makes the study quite a bit more interesting. And here's why. So these kinds of mutations just spontaneously occur, right? And natural selection hasn't had a chance to respond, or more accurately, the consequences of the mutation haven't had a chance to play out, either making the organism more or less likely to reproduce, right? That's a natural selection. It makes it so that the study is focused specifically on the process of mutation itself, not the broader evolutionary consequences of mutation. And so it's specifically asking, is every gene, every point along the genome, are they equally likely to undergo mutations, or are certain parts of the genome more or less susceptible? Or put another way, are certain parts of the genome protected against mutation? Okay, so this is a big study. It's in Nature after all. But, the main event, mutations do not appear to be entirely random, or at least not entirely random in the Thale crest, right? The plant on which they focused for this study. It seemed that certain genes were, just more susceptible to mutation than others. And so, more interestingly, the pattern of genes that were less susceptible to mutation didn't seem to be particularly random either, right? It wasn't the genes that had negligible roles in their survival or reproduction, of the plant. It wasn't those genes, right? No. The genes that appear to be protected against mutation were the essential genes. So once again, just to drive this point home, this is pretty fundamentally unorthodox when it comes to, genetics and evolutionary theory, right? This is the standard perspective on genetic mutations, as we discussed, right? And their consequences would be mutations occur randomly, just as a byproduct of the programmed imperfection of the various little proteins that, conduct genetic expression, right? If you remember, all these little, proteins, they're error prone. And then the consequences of those mutations play out via natural selection, right? A mutation can be completely unimportant, which is likely usually the case. A mutation can be advantageous in that it results in an organism that's capable of reproducing more effectively, or a mutation can be disadvantageous, right? Making it less likely that the organism will reproduce. That's the standard. So, they just happen. And they just happen randomly across the entire genome. Each of our 20,500 genes in the human genome, or really the 3 billion base pairs of A's, T's, C's, and G's, each of those letters in the human genome is equally susceptible to mutation, and after those mutations occur, you either get a human that's more or less or equally likely to reproduce. Okay, but the findings of this study suggest that it doesn't quite work that way, and as the lead author says, quote, "I was totally surprised by the non-random mutations we discovered. Ever since high school biology, I've been told that mutations are random." And that's absolutely what I learned in high school too, when I didn't even think that I could become a scientist. But then the lead author continues. The idea of random mutation has been around for over 100 years in biology, and is something you hear so often as a student that it's easy to take for granted. Even as a practicing geneticist and evolutionary biologist, I had never seriously questioned the idea. Okay, so, I won't go through all of the figures in this paper, because there's a lot, and I try not to lean too heavily on its jargon, but there's one term that I think would make the content of this paper easier to digest, and so that is gene body. Okay, so I know that that's probably not exactly what looks like what comes to mind for most people when they think of a gene, but this is basically just breaking down the different roles that certain stretches of a gene play, certain sections, right? And so you can see from left to right, you have the promoter, exon, intron, and then stop, right? Those are all composed of the same types of A's, T's, C's, and G's, just in different sequences, or bases, right? Those A's, T's, C's, and G's are also called bases. Those are the same types of molecules that compose our entire genome. But part of the gene is responsible for encoding the protein itself, right? The blueprint of the building itself. Other parts of the gene are responsible for attracting all those little cellular machines, right? They get there somehow, and that section called the promoter has a lot to do with that. And so, these types of little cellular tools that are responsible for reading DNA into RNA, and then RNA to amino acids and protein, right? That's called the promoter region. All of those little cellular machines are attracted to that promoter region of the gene, right? Sort of like the address of a construction site, right? It's how the dump truck and crane and cement truck all know where to arrive to start building something, right? They need an address to arrive at the right place at the right time, right? That's what a gene's promoter is. Then a small part of the gene is the start codon. So it's really just like a short sequence in the genome that signals, where the cellular construction crew should start, right? They shouldn't start building, a new cement truck, right? They should start where the blueprints indicate that they should start. And so that's where the body of the gene is, right? That's the literal code of the resulting protein itself. That's the blueprint, right? In the absence of anything else. It's just like a blueprint for a building that's sitting on a desk in some architect's office, right? Without having the construction crew and management and all those types of things, a blueprint sitting on a desk won't make a building, right? But if a construction crew has zero instructions regarding what they need to build, you're probably not going to get a building either. And so, that's the gene body, okay? And that's the component of the gene on which we're going to focus for the main nugget of this study. And so there's one figure in the paper that I think pretty intuitively sums up their argument. And you don't have to be a scientist to sort of like grok what it's conveying here. This one I think sort of sums up their argument. And so I know there's a whole lot to this figure that's like totally inscrutable, but don't worry about the numbers or really the axes at all. Just note that, one side of each of those graphs is either upstream or gene body, okay? And you can see that those lines are either rather high or rather low. And on the top we see, upstream and gene body, and pretty much without exception, the line is lower in the gene body section of the graphs. And that's showing that when it came to mutations that they observed, they were quite a bit less likely to have happened in the gene body section. I don't know if you can hear my daughter in the background. She's exploring volume. But in any case, so, as you can see, pretty much wherever, gene body is, let's just focus on the top two, right? That line is much, much lower. And so what they're showing there is that the mutations that they observed, they were just quite a bit less likely to have happened in the gene body section of a gene, the blueprints, the actual genomic code of proteins. And so in other words, they saw that there was just a lower rate of mutation in the gene body, in the blueprints, right? So that in and of itself is interesting. But they went a step further and showed that not only are certain parts of the gene less likely to mutate, they also showed that certain genes were less likely to mutate. Okay. And so I know this isn't exactly the most appetizing figure in the world, but don't worry about understanding all the various things on this graph, right? Just focus on two parts of this figure that are low mutation probability genes, all of which are blue right here, and high mutation probability genes. That's sort of orange color over here. And so they are basically showing the genes that are least and most likely to have undergone mutation from what they saw in those lovely little flowers, right? Each of those sideways bars is a different category of gene. And so some of the genes are responsible for things like how the plant will respond to being exposed to something like salicylic acid, right? Aspirin, or a wound, like a broken leaf. But some of them are responsible for like some of the most critically, centrally critical functions for literally all forms of life, not just flowers. mRNA processing, translation, right? Peptide synthesis, gene expression, right? Those are the types of things that are happening in each one of us right now, all the time, while we eat, sleep, work, zone out while watching a movie, whatever. These processes happen in plants, in dogs, cats, and rats, right? And humans. And so if you break one of those essential functions, if it has a significant mutation, that's game over, right? If one of those genes mutate, it's like removing the battery or the axles of a car, right? Or maybe even just breaking the factory that produces those car parts in the first place. So in other words, they found two categories of genes, two broad categories, some of which were less likely to mutate in blue, and some of which were more likely to mutate in orange. And it turned out that the processes that are, that are only sort of important, right? They're all sort of important, right? But at a minimum, sort of important. The ones, those are much quite a bit more likely to undergo mutation, right? The genes that are absolutely essential, on the other hand, the battery, the axles, right, or the car factory, those genes were quite a bit less likely to undergo mutation. Okay, so as a lead author of the study says, DNA is a fragile molecule on average, right? DNA can, in an average cell, is damaged between 1,000 and 1 million times every day, right? As he said earlier. And that's the fundamental engine of mutation, those errors. But then he goes on to say, in genes coding for proteins essential for survival and reproduction, mutations are most likely to have harmful effects, right? Potentially causing disease and even death, right? And so, there he's getting to the reason that certain mutations aren't inherited, because they can't be passed down, right? And so, as I said, there's much more to this study, right? They go far beyond just like the fact that harmful mutations aren't inherited. Rather, that there's something that is preventing or making it just much less likely for these genes here in blue to undergo mutation relative to these. The high probability genes and the high probability genes were just sort of like kind of important stuff. You could take it or leave it. May be important sometime down the line as it continues to mutate, but right now, you can take it or leave it. You'll survive without it, right? Whereas these genes are the ones that, like, without these genes functioning properly, you do not get a plant, or you do not get a human. And so, based on the results of our study, this is another quote from him. So, based on the results of our study, we found that gene regions, especially for the most biologically essential genes, are wrapped around histones with particular chemical marks. We think these chemical marks are acting as molecular signals to promote DNA repair in these regions. And so that is the sort of mechanism that they think is responsible for distinguishing between these categories of genes and why it is that these are much less likely to mutate than these. And so, so that's super cool, right? That basically there is a physical biochemical mechanism by which these distinct categories of genes undergo mutations. Pretty cool. And to be clear, the mechanism that he brought up, the histones attracting DNA repair mechanisms, that's just one of the whole variety of cellular mechanisms that operate to detect and either fix mutations, genetic mutations, or fix the protein that results from a mutated gene, or just get rid of the mutated protein altogether, right? There is a DNA repair component to that. And so, that mechanism does fit pretty nicely into Darwinian evolution by natural selection, right? The process of gene expression is just that. It's a biomolecular process. And so, just like human behavior is subject to natural selection, right? So is the process of genetic expression. It all is. And so our cells have various mechanisms, right, that respond to these mistakes that are made by those little, molecular machines along the way. But they can only respond to so many mistakes, right? And so, for example, I live in Philadelphia, one of the larger cities in the United States. We have quite a few fire departments and ambulances and emergency services and so on. And so, generally speaking, if there's an emergency, in the city, you can expect one of those emergency services to show up pretty promptly. And even if there's like a dozen or 24 emergencies happening simultaneously in the city. But what if there are hundreds or thousands of emergencies all at the same time? And the emergency responders can only respond to so many incidents at the same time. And so as a result, at a certain level, those emergency responders, can only put out so many fires or respond to so many medical emergencies. And so there's a similar concept here. These systems that repair errors in gene expression can't be just omnipresent and perfectly effective, right? As a result, it may very well be the case that various life forms evolved ways to kind of prioritize their surveillance of these genetic repair mechanisms to specific regions of the genome, right? The regions that contain the most essential genes, the types of genes that are essential to survival and reproduction. And so that's pretty cool. And so, non-random mutation. But they were focused on this one cute little flower, right? What was it? I think it was this one. Yeah. And that's inherently interesting. But as self-interested primates, I'm sure we all immediately wonder, okay, but is this mutation dynamic happening in all forms of life, right? Are the most essential genes in the human genome also less likely to mutate, as is the case with this cute little flower? So Monroe says, quote, "We think it is likely that other organisms could also have non-random genetic mutations. We have actually been following up with our study by investigating this question in other species, and are finding results that suggest non-random mutation is not unique to Arabidopsis." So in other words, they think the answer to that question is yes, that this is the case for essentially all forms of life. And like a responsible scientist, Monroe isn't just like outright saying yes, that's what happens, but they're providing their hypothesis ahead of time, and they're almost certainly already coming to a close, to concluding, some experiments that they had going on simultaneously. I bet that that's what they have been doing. But using a form of life that's, just more complicated than that cute little flower. The thale crest, by the way, just as a reminder of what it's called. So, while this whole non-random, mutation thing was focused specifically on plants, with an obvious eye towards life in general, this does have some relevance to another biochemical entity on Earth, viruses. So viruses evolve just like, entities that are uncontroversially alive do, right? Many scientists argue that viruses are not technically alive. But maybe the most intuitive example of this process occurring, right now might be how it is that, variants of a very specific virus that's been rather rudely changing our daily lives for the past several years at this point, how it is that a virus like this is able to change over time to evade our antibodies. And so every single time that a cell in our body, in anyone's body, is infected by a viral particle, that kicks off a whole evolutionary event. Okay, and at this point, I'm sure basically everybody, is at least a fair bit familiar with the various steps involved in viral infection. Right, there's a viral particle of some sort, one that looks like, this one, for example, in this that's the first. The first thing is the viral particle, and it has those little proteins along its surface, right? That like the spike protein, for example, and that spike protein, which like all proteins, is composed of a specific series of molecules called amino acids, and those viral proteins can interact with our proteins on the surfaces of our cells in our body. And so different cells in our bodies have different proteins along their outsides, right? And so a lung cell will have different proteins on its surface relative to muscle cells or skin cells or neurons, right? And so that's why some viruses are respiratory viruses, which infect cells along the places we breathe, while other viruses infect other parts of our body, like HIV, attacking white blood cells. Respiratory cells have different proteins on their outside relative to the cells that are part of our immune system. And so, as a result, different viruses infect different cells in the body. And depending on which specific cells a virus can infect, you can have completely different pathological or disease reactions, right? Some viruses cause coughing and sneezing and difficulty breathing and headaches and so on, right? By now. While other viruses could just cause the immune system to essentially shut down. Right? Different parts of the body, different cells, different proteins. Makes sense, right? And so, as you can see in this super duper brief animation, that initial protein interaction between, the virus and the cell, that one right there, is what enables the virus to enter our cells, right? And then after that, it introduces its payload, its genetic information, which is then manufactured by a lot of the cell's own machinery, right? The same exact machinery that expresses our own genes. And so it's almost as though, someone's like from a Ford plant invaded a Chevy factory, snuck in the plants to make a Ford car in the Chevy factory, and now that Chevy factory is all of a sudden making Fords, right? The virus is replicated and replicated and replicated and creates tons more of itself, ultimately escaping from the cell and then moving on to infect more cells. Rinse, wash, and repeat, right? That's how the virus reproduces. I'm sure a lot of that isn't new for many people at this point, but what's important here is that specific step when the virus's genetic material is copied and replicated by our body's replication machinery. Every single time that happens, every single time that genetic material is replicated, there's always that same opportunity for mistakes to be made. There are opportunities for the byproduct of the virus that's being replicated to generate a slightly different new kind of virus, right? It's not entirely new, but just slightly different. So in other words, there are opportunities for mutation every single time that genetic payload is replicated. And so instead of an A, in a part of the sequence of the virus gets a G, in at one specific point along the genome. And so this is happening billions and billions of times right now as we speak. And most of the times those mistakes probably don't really matter all that much, but sometimes the mistakes that are made result in a virus that's different enough from the one to which we've all been exposed that the antibodies we all have from vaccination or prior exposure just don't quite recognize it. It's sort of like, if the security in a mall or a store or something has been alerted that a dude wearing a gray hoodie, red baseball cap, and blue jeans is planning to rob the store. And then all of a sudden there's like a whole bunch of guys showing up wearing gray hoodies, red baseball caps, and blue jeans that show up. So security is like 100% on top of it. They're watching every single one of them just to make sure that they catch them if they rob the store. But all of a sudden, among all the guys wearing those clothes, there's a dude wearing, shows up wearing slightly different colored baseball cap, right? A green baseball cap. Everything else is the same. It's just a green baseball cap. And he's just slightly different. And so the security doesn't pay nearly as much attention to that specific person with the green baseball cap because, after all, they're so focused on, the initial information that they received regarding who was planning on robbing the store. And so the more opportunities we give viruses in general, right, and certainly this current virus, the more opportunities we give it to replicate, the more opportunities it has to change the color of its baseball cap, right? To just mutate, to evolve. And so, it's a similar sort of situation with viral mutation. The vast majority of all the viruses that are replicating in the body are essentially identical. But every time a mistake is made when they're replicating, there's an opportunity for the virus to mutate, to change the color of its baseball cap and avoid the attention of our immune system, of our security system. And so that's why literally limiting the number of times that viruses can copy themselves as much as we possibly can is good for everyone. Not just good for a person who's exposed to the virus, right? And it's good for them too, but good for literally everyone. Fewer opportunities for the virus to copy itself, fewer opportunities for one of its progeny to generate some kind of mutation that results in something that's harder for our immune systems to fight. Okay, I know that was a bit of a digression. I understand. I just couldn't help myself. Just given the inexplicable, and at this point, it has to be intentional. Given the inexplicable misunderstanding regarding how it is that we fight viruses, it's got to be intentional at this point. But regardless, the point here is that if the findings of the initial study are replicated by other groups, it would pretty much change the way that we think about the fundamental engine of evolution, mutation, right? And it's pretty cool. So that's what I have for you. Kind of a long one, I know, and I went on a bit of a digression at the end there, but if you dig this kind of discussion of biomedical science, maybe you'll consider liking and subscribing and doing all that kind of stuff. Otherwise, thanks for watching, and I'll see you on another Sunday. Hey, Scott. Not a mutation, but my birth defect makes it very unlikely that I'll reproduce. Oh, that's interesting. Sorry to hear that. I mean, I know plenty of people aren't particularly interested in reproducing these days, and sometimes I understand. Fun fact about Philadelphia Fire Department. It was set up by Ben Franklin. That's right. And only saved buildings of the owners who bought a membership, which was shown as a small cast iron shield on the building. I didn't know about that. That's interesting. Your analogy are anti-vaxxers are the store owners who didn't want to pay for security workers. Yeah, pretty much. Or that's what I mean by it has to be intentional at this point, because it's just the numbers at this point are just so overwhelming that it's just, yeah, I think that it's extended beyond just somebody just hasn't been exposed to the right information or only been exposed to misinformation. Anyways, thanks for watching, everybody. Maybe you'll consider subscribing. Otherwise, I will see you on another Sunday. Thanks for watching.